This presentation is on aerobic respiration using glucose as a source of energy. In another video I'll talk about anaerobic respiration and the other fuels sources for respiration. So firstly, let's get into it with what is the main aim of respiration, and that is to produce this molecule here, ATP. Okay, so ATP is a ribose sugar with an adenine nucleotide base and 3 phosphate groups. Okay, so it's referred to as a phosphorylated nucleotide. Obviously, it's a nucleotide with more phosphate bases. So we need ATP to be broken down in a hydrolysis reaction. Let's underline that for us. Hydrolysis reaction. Okay, and hydrolysis reactions of ATP produce ADP and PI, inorganic phosphate. So this releases a lot of energy. So this energy is used in all processes such as active transport, cell division, endocytosis, exocytosis, synthesis of proteins, DNA replication, cell division, etc. It's used in no end of processes. It is known as the universal energy currency. So ATP is a universal energy currency. So there's 4 stages to this. The first one being glycolysis. So glycolysis takes place outside of the mitochondria. The mitochondria thing's in red, so that orange circle I've just drawn is outside the mitochondria. It takes place in the cytoplasm. Okay, the next part is the link reaction, which is in purple, and this takes place within the mitochondria. Okay, that purple thing I've just drawn there, that is where, this is the matrix of the mitochondria. That's where we'll see the link reaction take place. Okay, then we have the Krebs cycle, which is going to be in green. And the Krebs cycle also takes place in the matrix. So anywhere in there is the Krebs cycle, even within that purple cycle. So anywhere within here, the matrix is where we're going to see the Krebs cycle take place. And the last one is oxidative phosphorylation. And that takes place in the cristae of mitochondria between the intermembrane space and the matrix. Okay, so as you've seen here, I've highlighted that area. These are cristae protruding out of the inner membrane. So this is the inner membrane, that's the outer membrane, and between the two is the intermembrane space. Okay, so ATP is the product of respiration, and obviously we don't want this cycle to continuously be ongoing. Sometimes we'll need more ATP, sometimes we'll need less ATP. So that's why ATP is the end product inhibitor, end product inhibition. Okay, so ATP inhibits enzymes at the start of the process, such as glucokinase enzyme, which we're about to see in glycolysis. So let's get into glycolysis. Glycolysis has 2 main parts to it. So it's got phosphorylation and oxidation. So phosphorylation, oxidation. I've split it into a third part, the phosphorylation, and then the splitting of the product of phosphorylation, and then the oxidation of that. Okay, so first of all, let's look at phosphorylation of glucose. So that what we're doing when we're phosphorylating something is we're adding on phosphate groups. So over here we can see a phosphate group being added on. I'm going to annotate that for you as a phosphate group, so PI, inorganic phosphate. Okay, then how do we do that? So we start off with our glucose here, and then we use a hydrolysis reaction to break down ATP into ADP. This releases 1-phosphate, so triphosphate to diphosphate. 1 phosphate's released, and that's added onto the glucose. to make glucose plus phosphate. This remains a 6-carbon compound. Then we repeat that again, ATP to ADP, releases phosphate. Here are phosphate groups joined on the end, and this creates this molecule of hexose bisphosphate. Okay, so it's basically a glucose with 2 phosphates added on. Right, so we've phosphorylated that. We used the glucokinase enzyme as a catalyst for that reaction. And then we've got our product of hexose bisphosphate, okay? And now we need to split this down. So hexose bisphosphate is considered activated, okay? Because we've given it energy by adding on our phosphate groups. So we break this hexose bisphosphate into 2 triose phosphate molecules. So as we see, we've got a 6C hexose bisphosphate compound, 6-carbon compound. We split it into 2 3-carbon compounds called triphosphate. So 2 triphosphate. Finally, we oxidize this triphosphate. So oxidation means loss. Okay, so oil rig. Oxidation is loss, reduction is gain. So when we're oxidizing triphosphate, we're losing hydrogen. So we've got these 2 3C triphosphate molecules. They're both dehydrogenated by dehydrogenase enzymes, so the hydrogen is lost. We're producing 2N, and from that we need to accept these hydrogens. We know that hydrogens cause a low pH, which can damage the protein, mitochondrial proteins. We don't want to get damaged, otherwise we'll die. So we need to get rid of this hydrogen. So what comes along is coenzyme NAD. So 2 NAD, they can both take on 2 hydrogens each to become reduced NAD, 2 NADH. So dehydrogenation of the triphosphate produces 2 NADH. Also, because we are activated hexose bisphosphate, we know that these TP molecules have energy, and this energy is used to create 4 ATP, so 2 ATP each. So one triphosphate will produce 2 ATP, the other triphosphate will produce another 2 ATP, then we've got 4 ATP. That's done by phosphorylation reaction, joining inorganic phosphates to ADP. 4 ATP produced. Our end product there, after the dehydrogenation and the dephosphorylation, produces 2 pyruvates. Okay, that's our end product, 2 pyruvates. And those pyruvates can then go into the link reaction within the mitochondria. Here's the link reaction. So as we see here, pyruvate is actively transported. It's actively transported through H+ import channels in the mitochondrial matrix. So let's go back to the mitochondria here and let's illustrate what I'm talking about there. So to get pyruvate in, we're going to have to get it into the matrix, and that is an active process. So what we know about active processes is we've got a protein channel. It uses ATP. So I'm going to draw on here a protein channel in the bottom corner, as you can see just here. We've now got a protein channel. This is called a H+ import channel. Pyruvate H+ import channel, the key word, which allows the pyruvate to enter the matrix. Okay, so we're entering the matrix. Right, let's go back to the link reaction. So once we've got the pyruvate actively transported into the matrix, the mitochondrial matrix, here it is. So we're in the matrix now, and we've got a 3-carbon compound of pyruvate produced previously by glycolysis. Okay, so this compound is too large, so we need to remove one of the carbons. and that is decarboxylation. This produces CO2. Anytime you see CO2 produced, you know a carbon has been lost. So you know that the compound has got smaller. Okay. The pyruvate also releases hydrogen, which NAD once again accepts, become reduced NAD. That's dehydrogenation. This decarboxylation and dehydrogenation of the pyruvate produces a compound called acetate. It's a 2-carbon compound because we've lost that through CO2. Then we combine this acetate with coenzyme A. Okay, coenzyme A plus the acetate forms a product called acetyl-CoA. Now that acetyl-CoA can enter the next reaction, but there's one more thing. So we've taken, this is for 1 pyruvate, and we can see a link reaction here. It says the products are 2 carbon dioxide and 2 NADH. Remember, we produce 2 pyruvate at the end of glycolysis. So this reaction happens twice. So let's illustrate that here. Just trying to copy this all. Copy. Let's paste it down here. Alright. There we go. So the reaction happens twice. Okay, so 2 pyruvate enter. 1, 2. 1 NADH there, 1 NADH there, 1 CO2 there, 1 CO2 there. That produces 2 CO2, 2 NADH. These acetate groups, they combine with coenzyme A and produce acetyl-CoA. Right, now we've got acetyl-CoA. This is where we come to the Krebs cycle. So acetyl-CoA from the link reaction then turns into acetyl group. 2-carbon compound, the CoA enzyme goes away. Okay. One thing to notice here is anything in grey, so like this, NADH, FADH, we're looking at dehydrogenation. Anything purple, when CO2 is released, we're looking at decarboxylation, so carbon is lost. Okay, so this acetyl group combines with oxaloacetate, which is a 4-carbon compound, so 2-carbon compound and a 4-carbon compound. This forms a 6-carbon compound, citrate. It's called citrate. So now we've got citrate. This needs to be reduced to a 5-carbon compound, and carbon dioxide therefore leaves, decarboxylation, hydrogen leaves, dehydrogenation, producing this 5C compound, which you don't need to know the name of. This 5C compound then once again is decarboxylated, carbon dioxide is released, it's dehydrogenated, hydrogen is released and accepted by coenzyme NAD, forming a 4-carbon compound. This time we see it remains a 4-carbon compound, but it releases a phosphate group which binds with ADP to form ATP. So we formed 1 molecule of ATP. And once again, this 4-compound then releases hydrogen, which is accepted by coenzyme FAD, which can only accept 1 hydrogen. Okay, happens again. We remain as a 4-carbon compound and we get dehydrogenated again, releasing more NAD, but finally producing the oxaloacetate 4-carbon compound, which remains in the cycle. So then another acetyl group will come, and the cycle continues round and round, producing all these products. We've done it here for 1 acetyl-CoA. Obviously, remember, we produced 2 acetyl-CoAs in our last reaction. So this cycle happens twice. The whole thing happens 2 times. So let's write that on the here. 2 times. Okay. If we undo those steps, so we can just see the thing. What we are producing. We're producing, after the 2 cycles, 6 NADH, 2 FADH, 4 carbon dioxide and 2 ATP. This is one of the hardest to remember. There's so many little steps, but you've got to remember that the cycle happens twice, and this is what we produce after the 2 cycles. 1 cycle per acetyl-CoA. Finally, we've got these NADH and FADH. We've got 10 NADH per glucose from all the processes. We've got 2 FADH that we just got from the Krebs cycle. And now we need to create that ATP. We've created some, but it's not sufficient. We need to create much more ATP, and this is where we come to oxidative phosphorylation. Okay? Sounds hard, but it's really quite simple. So we know that these NADH, reduced NAD and reduced FAD both carry electrons and hydrogens. So when they get to this intermembrane space, which we highlighted here in the blue, we know that oxidative phosphorylation takes place there. So NADH and FADH arrive at the inner membrane, this membrane here. They then become oxidized. They turn into NAD and FAD. Okay, they're oxidized, they lose their hydrogens and electrons. So maybe we should draw that on here. So this becomes let's do a capital A, NAD, and this FADH, reduced FAD, becomes FAD. Okay, so they're no longer reduced, they are oxidized. They're back to their normal state. We can observe them like they were before. They can re-enter the processes. Okay, they can go back to glycolysis and accept the hydrogens and electrons. They can go back to the Linkert reaction. They can go back to the Krebs cycle and accept all of the hydrogens and electrons we need. Okay, now what we're doing with these electrons and hydrogens that we've just released, okay, we've got these ions, Fe would be Fe2 plus, electron carrier proteins, okay? So these are proteins with an ion-ion heme group within them. So these electrons travel through the ion-ion group, providing them energy. So all of these ions have electrons travel through them. Okay, and they gain energy from that. So electrons carry a lot of energy with them, and they give that energy to the ion-ion electron carrier proteins. Okay, this energy allows the pumping of hydrogen ions through the electron carrier proteins into the intermembrane space. Okay, so the electrons generate the energy for hydrogens to be pumped through. So now we have hydrogens accumulating in the intermembrane space. Okay, we get a lot of hydrogens accumulating here, which is not too helpful for anyone. The quantity of hydrogens in this space now is so great that there's a proton potential. Remember, we call hydrogens protons. There's a proton potential or a chemiosmotic potential. Okay, I think it's important that we write that down as a key word. Chemiosmotic potential. Right, so we've got a high chemiosmotic potential here. Okay, that was a really poor arrow. Let's put which means that hydrogens want to leave the intermembrane space. They're accumulating here, they want to move back here because there's a proton gradient formed, okay? As we know about all the assimilates, if you've got high concentration, they want to move to where there's a lower concentration, which is within the matrix here. Okay, so high concentration in the intermembrane space at low concentration here. So they need to move out. And they do this through ATP synthase molecules. Okay, they have a turbine sort of head on the end. And what happens is the hydrogens travel through these ATP synthase molecules, and as they do that, they provide energy for the turbines to move. Okay, it's very complex. We don't have to know how this works, but we just have energy created here by these hydrogens to turn the heads. Okay, and then hydrogens accumulate. Once again, they accumulate at the bottom here. Okay, so we've got hydrogens here, and we've also got electrons accumulating there as well, because we've not lost them yet. Something needs to accept them, and that something here is on the diagram, and it's oxygen. So what happens is the hydrogens and electrons combine with oxygen to produce water. Okay, and there's an equation for that, which I don't want to get wrong. It's 4 hydrogens plus 4 electrons equal plus 1 oxygen molecule produces 2 H2O. Okay, so let's just quickly write that down. 4E minus plus 4H plus that oxygen here. So all of that forms 2 H2O, which is a byproduct of respiration. Okay, that is oxidative phosphorylation in a nutshell. Just some things to recap here. Over here, where we was travelling through these electron carriers, we call that the electron transport chain. So these ions, heme groups within the electron proteins, form the electron transport chain. Okay, and when the electron travels through it, it becomes the Fe2 plus becomes oxidized to form Fe3 plus. Okay, they become Fe3 plus becomes reduced to form Fe2 plus. Electron carriers have a coenzyme that electrons pump proteins, so they pump these protons into the matrix in the intermembrane space. Okay, this forms a chemiosmotic potential, acting as the source of potential energy. Protons cannot easily diffuse through this lipid bilayer, so they remain in the intermembrane space. However, some do leak out. Okay, but the majority don't, so we ignore the proton leak. As protons pass down their concentration gradient through ATP synthase molecules, they cause a conformational change in the shape of the ATP synthase enzyme, allowing ADP and ATP to combine, which I didn't really illustrate here, which is the most important thing, is after all of this, ADP and phosphate becomes ATP. Okay, so the pumping of protons produces ATP. We have 10 molecules of NAD, they produce 25 molecules of ATP here. And the 2 molecules of reduced FAD provide enough protons for 3 molecules of ATP to produce. Okay, so the turning of this head, powered by hydrogens, produces ATP, so ADP plus P.